In Star Trek, characters carry a little handheld device called a tricorder that they can point at objects to analyze and identify them. When the show's writers cooked up the idea in the 1960s, it was purely science fiction. But a new paper in the journal New Pythologist takes the idea a step closer to reality. The researchers used a handheld device that looks a little like a ray gun to record how plant leaves on different Alaskan mountains reflect light. And it turns out different populations of plants of the same species, for instance, plants living on neighboring mountaintops, reflect light differently in ways that echo their genetic variation from each other. While trained biologists can usually walk into the field and identify species with their eyes, it takes expensive genetic analysis to reveal the populations, groups of individuals of the same species within a gene pool, that are so important for conservation and evolutionary research. In this new study, the researchers have shown that we can use light instead of DNA to define plant populations at a similar level of detail. This new method is a lot faster and cheaper than genetic testing, which could dramatically increase our efficiency at mapping and monitoring biodiversity. All living things contain DNA, and the more similar two organisms' DNA is, the more closely related to each other they are. That is true both between and within species. Your DNA is more similar to a chimpanzee's than to a dog's because we are more closely related to chimps, and your DNA is closer to your cousins than to a random stranger on the other side of the world. The same is true for plants. Even within a single species, there are variations in DNA from one population to another. Genetic research has shown that sometimes these variations appear at a very fine scale. For instance, plants from one species on one mountaintop can form groups that have slightly different DNA than the plants on a mountaintop just a few miles away. When populations split like this, that means that they are not sharing pollen or seeds with each other and are genetically isolated. Scientists study these differences in DNA to tell one plant population from another, but it is an arduous task. They have to collect the plant samples, store them, get permits to move them to the lab, then go through the many steps to actually sequence the plant's genetic code and compare them. It is a process that takes weeks or even months. In this new study, however, the researchers have found another method to determine how closely related two plant populations are to each other, one that could eventually be done almost instantaneously out in the field. This is where the ray guns come in. Spectroradiometers are instruments that measure how much light reflects off a surface and what wavelengths that light contains. The instrument itself fits in a backpack, and there is a handheld probe attached to a fiber optic cable that looks like a little ray gun. Agricultural scientists use these instruments to analyze the light bouncing back off of leaves to detect disease. But this new study revealed that the light bouncing off of leaves varies from one population of plants to the next. Leaves have evolved to interact with light, and these machines are recording differences in the light after photons have entered the leaves and been observed or bounced around based on different chemistry and structure. This instrument reads the visible and infrared light that bounces back off of the leaf, and that information can give a tremendous amount of information about the chemistry and structure of the leaf. The scientists found that from one mountaintop to the next, the leaves reflected back different amounts of light at different wavelengths. And once they sequenced the plants' genomes, they found that these differences in reflectances corresponded neatly with plants' genetic differences. That means that looking at the light a plant reflects can be a quick, reliable substitute for lengthy genetic testing for researchers in the field trying to determine if a population of plants is genetically unique.